Hello, everybody. So we are going to be talking about cell division and mitosis, which is a form of cell division, in this video. Here we go. So mitosis is a form of cell division that all living things are capable of doing. But especially for eukaryotic cells, we do them under very specific conditions or circumstances. So our bodies utilize mitosis for growth and for maintenance of our cells. They are a form of asexual reproduction. So asexual meaning not two partners are involved, only one individual is involved, or in this case, one cell is involved. And you'll see that happening in many of our single-celled eukaryotic cells. And in plants, it's a form of cloning. Okay, so mitosis, what you're going to end up with are two cells that are genetically identical to each other. Those two cells are called daughter cells. And genetically identical means that they have the exact same amount and type of DNA as each other. So if my original cell had 8 chromosomes, when that cell reproduces and I now have 2 cells, they will each have 8 chromosomes of the exact same kind with the exact same information. Okay, so if you look at the image over here, literally it's like the cell splits itself in half. It copies everything on the inside, it splits itself in half, and one half develops into daughter cell 1 and the other half develops into daughter cell 2, and they are genetically identical. One of the most important steps in the cell division is the replication of DNA. Because if I'm about to have two completely separate cells that have to be the same, I have to make sure that I've copied every single bit of DNA in the original cell so that I have enough genetic information to split in between two separate physical cells. Okay. So because we're going to have this equal separation of DNA, we must first copy or replicate all the DNA. So this is going to happen in the original cell, and it creates this short-term condition where the number of DNA chromosomes, or the number of amount of DNA in general in that cell is doubled. That cell will be carrying twice as much genetic material as it originally does, just long enough so that cell division can actually take place. So once all of that DNA has been replicated, it will be evenly distributed between the two daughter cells so that we'll have two new cells, but they both have the same information as the original parent cell. So when exactly do we go from having DNA to having chromosomes? Well, chromosomes are made up of DNA. They're DNA that have been folded into a particular shape. Now, in eukaryotic cells, chromosomes only appear before cell division. Normally, DNA is just loose and strung out in the middle of the nucleus. So here's what I want you to envision. I want you to envision that you have several balls of yarn and you left them out and you have a kitten. And you come home and the kitten has played with every ball of yarn. So however that ball of yarn has unraveled, wherever it's lying, whatever it's stretching in between, that's just how it is. That's what your DNA looks like normally in the cell. But if we're about to copy everything and then separate the DNA into two sets, that could make for a messy situation. That could make for DNA that breaks or gets torn or gets messed up somehow. So to make sure that everything gets copied properly and that everything gets distributed properly, the DNA gets folded up into these chromosome shapes. So in eukaryotic cells, DNA is divided into these linear individual shapes that we refer to as chromosomes. They're going to be found in the nucleus of the cell, and they are specific to the organism or to the species that that organism is a part of. Now, DNA can come in sets, and we use the term ploidy to talk about how many sets of DNA a particular cell or a particular species can have. So for most eukaryotic cells, especially humans, our DNA comes in two sets. So we refer to that condition of having two copies of each chromosome as being diploid. So that's a full set of chromosomes. And for humans, our body cells are the ones that are going to be diploid. So body cells, think liver cells, skin cells, brain cells, muscle cells, all of your normal everyday functioning cells. Okay? Now the other condition is referred to as haploid. And haploid is the not exactly the opposite of diploid, but it's half of the diploid condition. So diploid is sometimes represented by the symbol 2N. Haploid is represented by the symbol N. And it just means half a set of chromosomes. So in other words, one copy of each chromosome. Okay, so again, in humans, our diploid cells are our body cells, sometimes referred to as our somatic cells. And our haploid cells are our gamete cells, our sex cells. So think sperm and egg in this case. So I want you to think of a somatic cell as everything that's not a sperm cell or an egg cell, okay? So over here, what you're looking at is a picture of all of the chromosomes in a human cell. We have 23 pairs that are referred to as a karyotype. Now, this picture shows you two copies of each kind of chromosome, which is why our cells are 2N, our body cells are 2N. So when you have two copies of the same chromosome, we refer to that as a homolog, which is a short version for the term homologous chromosomes. So the chromosomes are the same, meaning they carry very, very similar information. They're going to be the same size and in the same location, but they come from two different sources. So one of these chromosomes came from your mom, and one of these chromosomes came from your dad, which is why you're a combination of the two of them. Now, when it comes to replication, normally these chromosomes exist as just one line, let's say. So if you think of a arm and a leg, just one of these is still technically a chromosome. So these guys have already been replicated. So let's talk a little bit about that. So when I replicate something, I'm making a direct copy of it. I'm duplicating exactly what I see. So ordinarily, before that cell goes through cell division, there's enough information to make one half of a homolog, which still has all of the genetic information in it. So these chromosomes are still homologous, but we color them in different colors to represent the fact that they came from two different sources. So replication is going to take place, and notice where I once had one chromosome, I am now going to have two chromosomes. Okay, so each section of that chromosome is referred to as a sister chromatid. So this is one sister chromatid, and then this is the other sister chromatid. Okay. Sometimes the terminology gets a little confusing here. So like I said, in DNA replication, we are duplicating or copying all of the information to be able to have two exact copies of that DNA. We call those two exact copies sister chromatids. And then through a process of steps, those sister chromatids are going to get separated and pushed into two separate cells. That's known as chromosome segregation, and it happens during cell division. So I know I've been alluding to cell division for a couple of slides now, but we haven't actually talked about it. But before we can talk about cell division, we need to talk about what the cell is doing prior to cell division. So the whole process, all of the steps that a cell goes through from the moment cell division takes place up until that cell itself is ready to divide again is known as the cell cycle. The cell cycle is made up of some distinct steps that we sometimes refer to as IPMAT, but we'll talk more about that. So I want you to pay attention to the picture first, and then we'll look at the words. So here I have my newly made cells. These cells have just gone through cell division. See this orange section of this circle? That represents all of the daily things that cells do. They're just going around doing their business. They're performing the functions that they are designed to perform. So if it's a liver cell, it's helping your body to detox and do all of those things. But they're also preparing to divide again. So this orange section is known as interphase. The cell spends about 90% of its life in interphase. The majority of its time, it is doing interphase. Interphase is the preparation for cell division, for actual splitting of the nucleus and splitting of the cell membrane, okay? So that division is actually known as mitosis, which happens right here. So notice we're only in mitosis for a very small amount of time, probably about 10% of the cell's time is spent in mitosis. So think of interphase as I'm preparing to divide while I'm doing my regular job, and now I'm dividing. And then the cycle just repeats itself, okay? So now let's write some notes. So interphase is where we spend the majority of time. About 90%. Mitosis is only about 10% of time. So interphase is made up of 3 stages, and they are known as G1, S, and G2, and that is the order in which they go. So we'll talk more about what happens in those stages in a minute. Mitosis, on the other hand, is actually made up of 5 stages. Now, a lot of us just say 4. Some people don't necessarily include a middle step, but we'll talk about that. So the mitosis steps are prophase, prometaphase, which some people don't necessarily include, metaphase, anaphase, and telophase. And then the last physical thing to happen is known as cytokinesis. Okay, so you need to know that interphase is made up of these 3 steps, and that mitosis is made up of these steps, and then lastly, there is a step called, or an event called cytokinesis. So starting with interphase, like I said, this is the step where the cells are doing their intended job. For a long time, we thought that cells in interphase did nothing, because that's kind of what it looked like. But then as technology improved and we could see more things, we realized that that's not exactly true. So like I said before, interphase is made up of 3 stages. G1, sorry y'all. G1, the G stands for gap. And in G1, the majority of the time is spent allowing the cell to grow. That's the main function of G1. Then S phase is synthesis. This is where DNA and centrioles are replicated. And then finally, G2 or gap 2 is the actual preparation for mitosis step. So once we get through to G2, we are now in mitosis. And the first stage of mitosis is known as prophase. So there's some specific things that start happening that you can actually see if you look at these cells under a microscope. And notice we have a cartoon version of this for you on the slide, and then a real version of this. So one of the first things that you notice is that chromosomes start to condense. They go from being all this long, stringy looking material into being these shorter, almost fatter, little rod-shaped things, okay? Then the mitotic spindle, which is a whole series of attachments, begins to form. And that's going to allow the chromosomes to move when they need to move, okay? So that mitotic spindle is going to form. You see that better on the cartoon version. So that would be here and here. And they actually start to separate from each other. They're going to end up on opposite sides, which we call spindle poles. Then our nucleus is going to start to break apart, and it'll start to disappear. Spindle fibers are going to start forming between the centrioles, and then we have microtubules that will begin to assemble, and they will attach to a portion of the chromosome known as the kinetochore. Think of it as the protein that holds the two arms and legs of the chromosomes together. So prometaphase is the step right after prophase, but just before metaphase. And some people talk about it separately, and some people include everything we're about to talk about just under prophase itself. So it just kind of depends, okay? So in prometaphase, the envelope has completely broken down. The spindle is going to enter the area that the nucleus once took up, and the sister chromatids of each chromosome is going to connect to a spindle pole. The kinetochore, which is found on either side of the chromatid, is where the spindles are actually going to attach themselves to the chromosomes so that when those chromosomes need to start moving, they are already attached to the thing that's moving them. I always think of it as fishing, where I'm using a hook and a line and I'm going to reel that fish in using this hook and line. So that's kind of what your spindle fibers do. They attach themselves to the chromosomes, and when the chromosomes need to move, those spindle fibers get shorter, and as they get shorter, they drag the chromosome towards them. So let's look at spindle formation in a little bit more depth, and we'll point out some of these areas that I've been talking about. So the kinetochore, I'm going to use my laser here, is a protein. See this dark disc-like image right here? That's your kinetochore. You're going to have two of them, one on one side of sister chromatid 1 and one on the other side of sister chromatid 2. Okay, so think of them as being on the left and on the right of the chromosome. So this region is known as the centromere, but the protein that the spindles are attached to are known as the kinetochore. So here are my centrioles. All of these lines represent my spindle fibers. So the spindle fibers attach themselves to the kinetochores, and notice this one, chromatid 2, is attached to the spindle pole over here on the right, whereas chromatid 1 is attached to the spindle pole on the left. So they can literally be separated in half, okay? Okay, so in animal cells, which we are made of, the centrosome is going to divide, and the two parts are going to move away from each other, okay? In plant cells, they have no centrosome, so instead, their spindle fibers assemble around the nucleus. Centrioles are only an animal cell thing. We don't see these in plant cells. Okay, so the next step is metaphase. And in metaphase, this is where we're going to start having some true movement a little bit, okay? So at this point, my spindle is fully formed. Those chromosomes are attached to a spindle. And my chromosomes are actually going to align themselves. They're going to line up in the middle of the cell, and that is known as the metaphase plate. So by lining up in the middle, the chromosomes have now positioned themselves in the best place so that they can be separated to the left of the cell and the right of the cell to allow the other sets of steps to take place. Okay, the next step is anaphase. So in anaphase, those spindles are going to separate the sister chromatids in the middle of the chromosome at that centromere point and literally pull them to the opposite spindle poles. So if you look at the image, you can see this happening. All of these chromosomes are moving in this direction. All of these guys are moving in this direction. The cartoon picture actually shows it a little bit more clear, okay? So the only thing that's happening, and I say only, but it's a very important step in anaphase, is chromosome separation or chromosome segregation. They're moving to opposite ends, and this is going to happen until the middle is pretty much empty. Okay, so each sister chromatid is at this point considered an independent daughter chromosome because it's these individual chromatids that will be placed into the two new cells to start the process all over again. So at anaphase, there are two separate movements happening here. And this is one of the reasons why we started off the course the way we did, where we're talking about certain materials. Like, you already know what a microtubule is, okay? These things are not foreign to you. This is where it just gets applied in a bigger sense. So the kinetochores are going to move along kinetochore microtubules. This is actually going to pull the chromosomes to the poles, okay? And then the non-kinetochore microtubules are going to slide over each other, and that actually moves the poles further and further away. This two-step movement is going to ensure that my chromosomes end up where they need to be, so that when that nucleus actually divides and when the cell later on divides, I don't lose any chromosomes in that process. Think of it as pushing you to the edge so that we can make sure there's plenty of space to form cells around you and nuclei around you. Okay, the last step is telophase. And I've always thought of telophase as prophase in reverse. So in telophase, my chromosomes are going to decondense. They go from being chromosomes to being more of the typical look of your DNA. Okay, that extended, relaxed, string-like image is what's going to happen. And then around these chromosomes, a new nuclear envelope is going to form so that I now have a nucleus again. Okay? Then, so at this stage, the telophase cell is pretty big because we're about to split the cell into two. So we've just set ourselves up to have two cells. If you look at the picture, you can see here I'm starting to make a nucleus over here, starting to make a nucleus over here. And then in the middle, I have this weird, like almost like a number 8 shape taking place, right? So that groove in the center is referred to as a cleavage furrow, which is what it's called in animal cells. In plant cells, it is referred to as a middle lamella. And it's this thing that's actually going to allow for the splitting of the cytoplasm, and we'll end up with two separate cells. But telophase is the last step of mitosis itself. The rest of it, what we're about to talk about, is just dividing the cell. Mitosis is the division of the nucleus. So once that nuclear envelope starts to reform, that step has been completed. So cytokinesis is the very last step in cell division itself. And in cytokinesis, it's the division of the cytoplasm. So this step is what actually produces my two cells. Because if you think back to that picture of telophase, it was still one big cell because it's all connected by one plasma membrane, okay? Mitosis produces my new nucleus for each daughter cell, but cytokinesis splits the body of the cell in two. In animal cells, which is what you and I are made up of, this step is preceded by furrowing. So that grooving that I pointed out to you on that telophase picture, that's what furrowing is. And it's just these bands of microfilaments that form under the plasma membrane, and they just contract. And it's almost like they constrict the cell and cut it in half from the inside. So here's a picture of cytokinesis via furrowing. So let me switch to my laser. Here are my microfilaments. And as those microfilaments grow, literally they push the cell inwards. So see this arrow here is telling you this part of the cell is going this way. This part of the cell is going this way. All of these parts are going inward. So, because don't forget, a cell is a 3D thing. So I constrict and cut myself in half, and now I have my two separate cells. Now in plant cells, it happens a little differently because plant cells have that pesky cell wall that they have to accommodate for. So the cell wall material is actually deposited along the former spindle midpoint, so the middle of the cell, we're going to start laying down this new material that's going to turn into the cell wall. And that laying down or that deposition is going to continue until a whole entirely connected new wall is built. And that's going to separate the daughter cells, cell 1 from cell 2. Okay, so this starts off as laying down the cell plate. And once that happens, it's going to form the middle lamella, which is the cells that will actually secrete the new cell wall. Okay, so this image shows all of the cell cycle. So starting off with interphase and into prophase, where we can start seeing the condensing taking place. Then into metaphase, here's all of my chromosomes in the middle. Here are my 2 spindle poles. Anaphase, where they've moved away. You can tell they've moved away because the space between them opens up. And then lastly, telophase, where we're going to start to lose some of that condensing and rebuild our nuclear envelope. So the next question comes up with how is this whole process controlled? Well, the cell cycle is a highly regulated process. And while there are many parts and pieces to this regulation, we're going to focus in on some very specific ones. So there are proteins and enzymes involved in the regulation of cell division. And they are known as cyclins and cyclin-dependent kinases, or CDKs. They are the internal controls that are going to control how cell division moves ahead. Okay? So along with these cyclins and CDKs, we also have these internal checkpoints. They don't happen in every step, but they happen after a lot of major events. So in some cases, you're checking for multiple things at once, but we'll talk more about those. So what a checkpoint does is they will stop the cell in the cell cycle if something has not been done properly or all the way through, or something is just incomplete or incorrect. Okay, so they pretty much ensure that every stage is completed before the cycle moves on to the next stage. So there is a checkpoint in G1, and it is the main checkpoint that's pretty much determining whether or not we can continue to grow. So that G point checkpoint is checking for, does my cell have enough nutrients? Is it growing properly? Am I getting big enough? Is there any initial DNA damage? Okay. That growing part is really, really important, to the point where a lot of people think that the main focus of G1 is to grow. And it kind of is, because if I'm about to split in half, I need to make sure that I'm big enough. I have enough volume to split between 2 cells. If I split a tiny cell into 2 tinier cells, there's a good chance that they will both die. Now in the G2M checkpoint, this is where we're pretty much committing to cell division at this point, okay? But really what we're checking for in this G2 checkpoint, again, we're checking for cell size, because at this point you've been growing and growing and growing, so you should be big enough. But more importantly, we're also checking for DNA replication. Did all of the DNA get copied? Because the goal of this is to make sure that I have 2 genetically identical daughter cells. And if I haven't copied all of the DNA, then they can't be genetically identical. And then there's an M checkpoint, and it actually happens in metaphase of mitosis. And we are checking to make sure that the chromosomes have attached themselves to the spindles properly. Because if they don't, then chromosome separation doesn't happen the way that it should. Okay, so let's go back and talk about those internal controls again. So like I mentioned before, we have proteins and enzymes involved called cyclins and CDKs. Those 2 are capable of attaching themselves to each other and forming complexes. And they directly control the cell cycle. So CDKs are activated when they are combined with a cyclin. So as cyclin levels increase, CDKs get attached to them, and then that tells the cell cycle, hey, you're free to move on to this next stage, okay? That attachment adds phosphate groups to specific target proteins, and that activates the protein in turn. These activated proteins then trigger the cell to move on to the next stage. So no cyclin, if cyclin levels are low, CDKs do not attach, phosphate groups do not activate proteins, and proteins do not trigger the cell to move to the next stage of the cell cycle. So those are the internal controls. External controls also exist, okay? And they help to coordinate mitosis with everything else that individual's cells or bodies are doing, okay? An organism cannot have every single cell go into mitosis at the same time. So while some are doing everyday living, a few of them are going through mitosis. Like it's a constant back and forth between who's dividing versus who's not dividing. So how is that coordinated? It's based on proteins or surface receptors that can recognize and bind signals, bind messengers pretty much that come in and say, hey, this cell's dividing versus this cell is not dividing. So those messengers come in the form of hormones, protein hormones, and also growth factors, which are also types of proteins that tell the cells, hey, it's time for you to go ahead and divide, or hey, don't divide just yet, okay? And once these hormones come in and they bind to the cell surface receptors, that is going to trigger the internal controls that we just talked about to either speed up, slow down, or stop cell division altogether. So a cell and an organism in general isn't going to put all this effort and energy into controlling a process if it wasn't vital or essential. And what happens when mitosis is not controlled is cancer, okay? So when you end up developing the condition of cancer, it's because cells have lost control of cell division. The regulated steps no longer exist. So these cells just divide continuously and uncontrollably, more than likely before they're completely ready. And this is going to create a growing mass of cells that stops your body from functioning in the way that it should. Okay? There are times where cancer cells break free from their original tumor. We call that metastasize. And then those groups of cells can travel throughout your body, in your bloodstream, and form additional tumors in other parts of your body. Okay. So as you know, cancer is a common disease. It's very often lethal, and it affects many, many people and even animals. Regardless of the different forms of cancer, all cancer cells derive from cells that blow through their cell cycle checkpoints. Their checkpoints have failed. The checkpoints didn't stop them, even though all the steps necessary were not completed. Okay? So tumor forms when one or more cells begins to divide uncontrollably. And there are two types. So benign tumors are non-invasive. So a lot of people tend to think of them as, tumors that you don't have to worry about, whereas malignant tumors are invasive and can spread throughout the body, utilizing either the bloodstream or the lymphatic system. And when they spread, they can start growing in new places. Right, so here are some examples of what tumors look like. So of course, here is your real life picture. So just giant mass of cells that aren't actually helping the body. They're not doing any of your body functions. They're just growing and feeding off of your nutrients. Here's another example of a tumor mass. And if you've ever heard of the term HeLa cells, so HeLa cells are a immortal line of cells. They don't die. And they were first harvested from a woman named Henrietta Lacks, who had cervical cancer. Her cells were taken without her knowledge or her consent, and we've used these cells for decades in science. HeLa cells are at the basis of some of our most important scientific discoveries, especially in terms of medicine and how cells respond to medicine. So the cells were named Gila after her. They weren't really named after her. That was the code name based off of her name, but eventually the story of her and her cells and their importance to science became known to the general public. Okay, so what about cell division in prokaryotic cells? Because this whole time we've been talking about eukaryotic cells. So in prokaryotic cells, cell division is referred to as binary fission. And it's a very similar process to mitosis. Where it starts, though, is a little bit different. So first of all, bacterial DNA is not linear. Bacterial DNA is circular. So let's write a note about that. So because of this, they don't truly have a start or an end point that you can easily see, not like with our linear chromosomes, okay? So there are places within the chromosome of a bacteria that we call an origin of replication, also known as an ORI site. So that's where replication is going to take place, okay? So think of it, if you look at the picture, you can kind of see right here, it just kind of opens up like a bubble, and then it moves all around in both directions until all of the DNA has been copied. Once all of the DNA has been copied, once it's been duplicated, you're going to have two full sets of DNA. And then those cells are going to grow a cell wall in the middle, kind of like plant cells, and then they'll separate into two separate parts. Okay, these pictures show you mitosis in animal cells versus plant cells side by side. For the most part, they look pretty similar. All of the steps are pretty much the same. It's really that laying down of the cell plate and growing the middle lamella and finally dividing the cell in half via that cell wall, as opposed to forming a cleavage furrow and pinching the cell into two separate cells. That's different. Now, as always, the cartoon images make everything look nice and neat and easy to see and find. But in real life, real cells are messy, so they may not look exactly like what the cartoons look like. So here we went through and picked out a couple of cells that are in all of the different steps of cell division. So here's my cell in interphase. I can't even really see my nucleus or anything. My cell is just doing its normal thing. Okay, so cell 2 is doing prophase. You can start to see it condense. You can see the outline of the nuclear membrane. Cell 3 is doing metaphase. Here are my spindle poles, and here is my chromosomes in the middle. Cell 4 is doing anaphase. Those chromosomes are starting to move away from each other. The space in between them is opening up. And then lastly, cell 5 is doing telophase. And if you follow the membrane, you can see that it's still all connected. But here my chromosomes are getting ready to have the envelope reformed around them. Okay, and now this image shows you mitosis in plant cells. So again, here we have our cell in interphase. This is our cell in prophase. We know because we can still see the nucleus around it. Here is our cell in metaphase. They're lining up in the middle. Anaphase, they're moving away from each other. There's space opening up in between them. And then our cell in telophase. And again, look at the membranes completely surrounding both of those clumps of chromosomes. So it's not split yet. Okay, so let's do a quick side-by-side of mitosis, which happens in eukaryotic cells, versus binary fission, which happens in prokaryotic cells. So somatic cells or body cells that are eukaryotic do mitosis, whereas prokaryotes do binary fission. The goal of mitosis in eukaryotic cells is for growth and maintenance and repair, whereas in prokaryotic cells, this is their form of reproduction. The nucleus is going to divide just one time. In binary fission, it's also just going to be a one-time division. The chromosomes are linear, and in binary fission, the chromosomes are circular. The result is going to be two genetically identical daughter cells. for both mitosis and binary fission. And in animal cells, or sorry, in eukaryotic cells, this can take anywhere between 24 to 48 hours, versus in prokaryotic cells, it can take anywhere between 30 minutes to 24 hours. And then lastly, sorry about that, y'all, the 4-year-old's up. Anyway, so the last thing I was going to say is that in mitosis, if we lose control of the process, it results in cancer. All right, and that's that for this video.